Well, good morning, everyone. Welcome to BSc 1005, or the Introduction to Biology for Non-Majors. As you've noticed, we are not meeting in person, which comes with a lot of weird caveats and things. So I hope you all will kind of bear with me through some of this stuff as we get going through here. Just as a brief introduction, and my name is Matt Atkinson. I'm a PhD candidate in the Savage Lab here at UCF. And we'll get more into my background at some point later, but this class is being taught asynchronously, which means that it's going to be a little different than what y'all have dealt with before. Basically, once a week, I will post 3-ish lectures. It depends on, holidays and stuff, as well as when we're taking exams. And you'll be asked to watch all 3 lectures, and then you'll have a weekly quiz. And we'll get more into all that stuff in a second. So let me go ahead and pull up the, sorry, words are escaping me. Let me go ahead and pull up the, all right, great. Now that I've got the PowerPoint pulled up, let's go ahead and get started here. So let me scooch this out of the way so you guys can still see me and still see that I'm a real person, but we'll start kind of going through the syllabus and things, and then we'll go ahead and get started on our first lecture. So again, my name is Matt Atkinson. I'm a PhD candidate, which means I don't quite have my PhD yet, so doctor doesn't quite work for me yet. I have my master's and my undergrad. I did my undergrad at University of Georgia, and I did my master's at the Western Carolina University up in Cullowhee, North Carolina. And I've been teaching here at UCF for about 4 years. So I've got quite a bit of teaching experience, but this is my first time actually teaching a real course. So I might have been a lab TA or something like that for the last, say, 6 years or so, but this is my first time actually running the lecture. So give me a little bit of grace. There will be times where I'm still kind of learning things. So I'm primarily what you call a herpetologist. So if y'all remember Steve Irwin, I kind of do very similar things. So I work with reptiles and amphibians. Primarily, I work in the disease ecology side of things. So frogs are actually probably one of the worst hit groups when it comes to pathogens. So that's something kind of interesting. And it kind of gives me some interesting insights onto this whole COVID-19 thing. And just so you all know, I've got an entire lecture saved up for disease ecology towards the end of the class. We'll be talking about everything from COVID-19 to the worst disease known to man, better known as the chytrid fungus, and a couple of other things. So should be fun, but we got to get through all the other stuff first. So I've worked with a bunch of really cool animals, everything from alligators to snakes to turtles, but I'm currently working with frogs for my PhD. So if you need to get a hold of me, please use this email and this email only. It is matthew.atkinson@ucf.edu. Give me about 24 hours to respond or so, and give me a little bit of leeway, especially over weekends and holidays, because, I have a family. I have 2 dogs and all that other kind of fun stuff that also has to take some precedence in my life. So give me a little bit of space on that one. And again, if you could please make sure you're emailing from a knights.ucf.edu account or through web courses, so that way I know you're not just some random spam and it gets caught in my junk folder. That would be fantastic. So office hours are going to be a little funky as well. They're going to be primarily focused being entirely online, and ideally I'd like to schedule them as needed. I am working on maybe setting up like a workshop once a week where we can open up a big Zoom meeting and have a bunch of people pop on and ask questions and all that kind of fun stuff, but I haven't totally set that in stone yet, so bear with me on that one. So obviously, this is an asynchronous course being taught online. One of the most important things you need to have is internet access. You're going to need it to get onto web courses. It's going to be where I'm going to post slides. It's going to be where I post the lectures, videos, and all that kind of fun stuff. So please make sure that you are paying attention to that. Go ahead and set up your web courses to where you'll get notifications every time I drop new files or put any announcements in there, because that'll be really helpful for you. So I've already gotten a couple of emails about this, but I'm not requiring a textbook per se, but I do really recommend this Biology, The Essentials 3rd edition. It's pretty thorough. It'll get the job done. And specifically, I really like this version of it that's online that the UCF bookstore sells, and I'll talk about that in a second. And it's called the Smart Book. It's really helpful for studying. I'll let you all decide if you want to do that or not. It's totally up to you. So the way you can get to that is that mheducation.com, and it'll give you access to that. You can just buy the book on Amazon, or you can get it through the bookstore. Totally up to y'all if you want to get it or not, but I really recommend it. It's going to help you study, and hopefully you're not going to be totally lost in this course. So, totally up to you, but I really recommend it. Here's a little bit of an example of what that Smart Book stuff looks like. So you can see, say here's a question that it'll prompt you. So inherited traits, characteristics, or behaviors that will enable an organism to survive, reproduce, and successfully in its environment are called what? And then at this point, you'll be able to click whatever answer you think is right. It's probably adaptation. And it'll tell you, oh, you put zygote. Whoops, sorry, that answer isn't correct. It'll tell you what the correct answer is, and then it'll explain to you why it's the correct answer. And then it'll actually, you can go through and get to these highlights section where it'll say, hey, this is the section you seem to really know well. This is a section you could use some practice with, and this is a section that you really don't know anything at all. So it has its benefits that can be pretty useful if you like those sorts of things. I'm a little bit more old school in that regard, but we'll get to that in a bit. As far as grading breakdown goes, A is 90% or more, B is 80 to 89%, C is 70 to 79%, D is 60 to 69%, and F is anything less than 60%. Honestly, this isn't going to be a hard class, or at least I'm not going to try to make it difficult and go out of my way to do things like that, but there will be some more difficult subjects that some of y'all may struggle with, but this is pretty much set in stone. So I'm pretty sticklers when it comes to some of this grading stuff. As far as your assignments goes, there's only really two assignments. You have weekly quizzes, which you're going to have 14 of. They get roughly 33 points per assignment with a maximum of 400 points. So 40% of your grade is going to come from these weekly quizzes. And then you're going to have 4 exams. I don't drop any of them, but you have 4 of them, and they're all very, very different. So say exam 1 is going to purely focus on just the introduction to biology and some of the chemical stuff. Exam 2 will be all about genetics. Exam 3 will all be about system biology, so talking about, anatomy and physiology. And then Unit 4 is going to be all about ecology. So they are very, very different, and that's why I really believe that you almost have to have access to, or be able to be tested on all 4 of those things. So sorry about that, but I'm pretty strongly firm on that one. At this time, I don't anticipate any extra credit activities, but give me some time. I'll think about it, and maybe we can try to incorporate something that will be fun. But I'm pretty strict about making sure that, only those that put in the extra effort might even get options for extra credit. So don't come to me at the very, very end of the semester and say, hey, I haven't been doing anything in this class all semester. Can I get extra credit? That's not how this is going to work. So getting to your exams, you're going to have 4 exams. Each exam will cover a specific unit. So i.e. exam 4 will focus on all the lectures related to ecology and conservation. My plan is for each exam to be limited to about 60 minutes, and they'll open at 12:01 a.m. the day of the exam and close at 11:59 that same day. So that should give you the entire day to be able to take it. So obviously with this being asynchronous, it's a little bit different of a class, but just something to think about. Big things. I'm going to try to get it set up to where you'll take this in one of the cheating prevention browsers, but I'm not going to go out of my way to say you have to go take this in the testing center. But the big things are, I don't want you using a cell phone. You won't be allowed to talk to other people. And if you get caught cheating, you're going to get a Z grade, and we can talk more about that. All that information is in the syllabus. Please make sure you read through it. When it comes to weekly quizzes, there's going to be 14 total because there's 14 weeks. Each quiz is going to be limited to about 30 minutes, so be sure that you have done all of the reading and listen to the lecture before you even attempt them. The quizzes will be available at the beginning of the week and will close before the beginning of the following week. So say the day that I post all these lectures will be Monday, August 24th. It'll close by the start of August 31st, which is that following Monday. And you'll have, those 7 days to take that quiz and, watch all those lectures and all that kind of fun stuff. The quizzes will be open book, but you're only allowed a 1 attempt. So, and your 2 lowest grades will be dropped at the end of the semester, i.e. you're going to be scored based on your 12 best quizzes. As far as the excused absence goes, see the list for reasons for excused absences. Again, this is an asynchronous course, so it's going to be pretty hard to have a good, real excuse. Now, if you're sick for a couple days, I totally understand, especially towards the end of the week where it might interfere with the quiz stuff, but make sure you have a legitimate excuse, and that can be health, religious observances, court appearances, official UCF or sports-related events, military deployment, a bunch of other examples of excused absences. Those are all in the syllabus, so please make sure you look at those before you start talking to me about that. That's just a good general rule. If you have questions, look at the syllabus. If the syllabus doesn't answer it, come talk to me. And specifically, make sure you provide those excused absences within 1 week of absence. That's something that UCF requires. So please just do that for me. It'll make all of our lives easier, especially with this being a very, very large course and it being my first time teaching a real lecture course. I hope that y'all can kind of give me a little bit more leeway on those things and make sure if you do have an excused absence, you're turning in those correctly. Obviously, with everything going on with COVID, this is going to be a little bit more of a complicated situation. So do what you can to be patient, and we'll try to make sure we get everything squared away. I understand, I've got very vulnerable family members that are important to me that, if something were, God forbid, to happen, I'll try to make sure everything goes up and according to plan. But, there may be a day or two where even I miss something. So I totally understand with this whole COVID thing. Just, we're all going to try to give each other a little bit of grace and work with each other and show a little bit of patience, and life is going to be good. So, one really important thing that you need to make sure you do, take the participation quiz. This is going to allow you to get your financial aid. Please take it. It's something the state requires. It's really weird. No other states that I've taught in requires this, but it's something that the state requires, so that way we know that you're actually participating in the course. You have to do this by Friday. So Friday, August 28th, make sure you participate in the participation quiz. And it's going to be a really stupid, easy answer. You're not going to get, this isn't your weekly quiz per se, but I do need to have this done by Friday. So, some things to think about. All right. Now, remember, if you do have any questions, please feel free to email me, again, at matthew.atkinson@ucf.edu. And again, remember all those things that I discussed earlier about making sure it doesn't get caught up in my spam inbox, because unfortunately, the UCF emails do have a good filter for those things. So you need to make sure you're emailing from a Knight's email or through web courses and all that kind of fun stuff. All right. So let's go ahead and get started on our first actual main part of the lecture. It's simply just, what is science? Which is kind of a very, very loaded term. And, thinking about this whole situation that we're in with COVID-19 right now, you hear the word scientists thrown around and science and all this stuff, and science got it wrong. And unfortunately, that's kind of a bad misconception of what exactly is science, and it can cause a lot of problems, especially when you're trying to make sure that everything comes out right. But first, what is a scientist? Now, I'm sure a lot of people, particularly those of y'all that aren't super familiar with interacting with scientists, think of this kind of being your stereotypical, scientist person. Somebody writing on a clipboard with a white coat and, the stethoscope around. Honestly, that looks more like a doctor to me, but most scientists don't look like this. Most scientists don't talk like this. Most are pretty average, great human beings that are regular humans too, and just have an interest that isn't, business or whatever. So here's a little bit better of an image that I like to show that shows what science is really like. So in that top left-hand corner, you have me with some of my undergraduate students at a conference recently. Not all scientists look alike at all. We come of all creeds, all races, all colors, all sexual orientations, all genders, all that kind of fun stuff. So a scientist is not just a boring old white man in a lab coat. And from that, you can get some, and scientists aren't always stuck and pigeonholed in the labs either. Here you see on that top right-hand corner, me working at one of my field sites. It was probably 11, 12 o'clock at night, and I'm sitting there trying to swab and handle some of the frogs so I can find out how much disease they have at a given area. And funnily enough, scientists aren't just all boring old fuddy-duddies that get stuck in their buildings all the time. That bottom picture is me and my boss, my PhD advisor, Anna Savage, building a fire because we got trapped in a canyon in the middle of Arizona searching for frogs, and we needed the fire to scare away actual cougars that were not too pleased that we were in their territory. So scientists can look very, very different and do very, very different things than what you normally associate with science. I really like to point that out. Now, the big thing that does unite scientists is that primarily science is focused around using this scientific method. So in general, all scientific inquiry is going to follow through this standard process to study the natural world. And you've probably been exposed to this in high school and stuff, so kind of bear with me here. I'm sure that a lot of this isn't new information to you, but do make sure you understand it. Now, know that this process is called the scientific method. And it's not a purely circle. This isn't something you do every single time to the exact T. There's going to be little side shoots, and there's going to be times where a project that you think is going to work perfectly, and you run the experiment, and the experiment fails totally, and you have to go back and say, okay, why did this happen? But it's often in those failures that we find some of the coolest stuff. One of my favorite examples of this is the way we discovered penicillin was because somebody who was studying, I believe, bread fungus accidentally left some out overnight, and it colonized one of his plates, and next thing you know, it killed all the bacteria on his plate. So there's a lot of really useful things that come from these weird accidents. So that's just something to keep in mind. And remember, again, these all have many interrelated paths. So usually, you make an observation. So say, for instance, in this case, rotavirus is common and causes serious illness. Vaccines are often cost-effective way to prevent illness. So you're going to ask a question like, is the rotavirus vaccine effective? You're going to then pull in your prior knowledge and formulate a hypothesis. You'll then make predictions using that hypothesis. Notice a hypothesis doesn't always directly mean that it's a prediction. And then you're going to design an experiment around that, collect and interpret the data, which honestly, in the grand scheme of science, the thing that you think the most of when it comes to science is actually a very, very small part. You're going to then use that information, use prior information to then draw conclusions from what you gather. So say, for instance, you're catching alligators, and there's one particular pond that has a bunch of smaller alligators. And you didn't, measure, you didn't determine what sex they were or anything like that, how old they were, anything like that. But just based off of prior knowledge and the knowledge or the data that you collected, you could probably infer that this pond is mostly consisting of younger individuals because they're all relatively small. Little things like that. Now, ultimately, you want to get to this point where then you start to publish that information. Because the big thing is information in science doesn't mean anything unless you publish it, which can be kind of frustrating sometimes because there's quite a lot of little walls and all that kind of fun stuff. So basically what happens is you write up the manuscript, and that usually means you're going back and forth between you and a bunch of different co-authors. It's constant editing for like months on end. You finally get it all pretty. You send it off to the editor for a journal, and they send it back with so much red that you would think that they killed something. And then you go through that process again and again, and finally you get that one paper out. And it can take sometimes years just to go from the point of collecting data to actually getting that published. I still have a study that I did in my master's that I finished in 2015 that is still yet to be sent to a publisher because we're still in the process of going back and forth between me and a couple co-authors. So it's a very long and labor-intensive process. Again, to kind of go back through this, the scientific method begins with observations and questions. So we talked about this rotavirus example. Another great example could be, you notice that a lot of, say, alligators in this one particular area look a little bit different. Maybe they have a little bit more yellow on their snout. Maybe they retain their juvenile coloration later in life. Who knows? And so you ask the question, what's causing this differentiation in color in alligators? Is it something genetic? Is it something in the environment? Who knows? So you then look back at prior knowledge. So say we'll go look through all the literature on crocodilian coloration, and we'll see, okay, maybe a Chinese alligator, which is a different species, can get this weird coloration sometimes in certain environmental conditions where maybe they're exposed to certain heavy metals. So then what you're going to do is say, based off of my understanding with these Chinese alligators and the fact that we're seeing this weird coloration in American alligators, maybe it makes sense that these Chinese or these American alligators are being exposed to some sort of toxin in the water that's causing them to change their coloration. And so that's where you're formulating that hypothesis. Now, Ken Fedorka, who's another professor in the biology department, is very, very particular about these kinds of hypotheses. And he really thinks it should be created into this if-then statement. So basically it'd be something along the lines of, based on our prior knowledge of Chinese alligators and their ability for their coloration to change as a result of exposure to heavy metal contaminants, and the fact that American alligators are showing these very similar kinds of discoloration, we would expect that these American alligators that we are finding, their discoloration is a result of, heavy metals contamination. So then what we're going to do is we're going to go out into the field, and we're going to catch a bunch of alligators, and that's the fun part. And we're going to, say, take some tissue samples to be able to look at and, check to see if they are being, exposed to heavy metals contaminations, or if it might be just some weird genetic abnormality. So we'll also, take some genetic samples from those same animals. And then we're going to go through and look at all the genetics and compare our weird little population of American alligators to the rest of the American alligators. And say, for instance, in this case, which is often the case, our hypothesis is totally wrong, and it's just some sort of weird color abnormality that's caused by genetics and has nothing to do with, exposure to lead or something like that. We can then pull that other information and say, oh, well, we didn't see any increased level of lead exposure in this population, but we did see that the genetics are different from this population compared to another population, say 10 miles down the road. And so from that perspective, we found that this coloration change is a very small change in the genetics. And that was the conclusion we drew from that. Then we're going to send that off and write it up and all that kind of fun stuff. And again, this can take years once you get even to this point. So, these projects are constantly ongoing and rather quite stressful, so it's just something to think about. And the big thing to keep in mind too is experimental design has to be very carefully planned, otherwise things don't work. So well-designed experiments include sample size, so the number of subjects in a group. And this is something that there's math that exists just to determine how big your sample size should be if you're trying to look for a specific amount of statistical difference between group A and group B. An independent variable. So this is the variable that's manipulated. In our instances, we'll use a different example. So, going back to classic genetics, you think of somebody like Gregor Mendel, who did his pea plant breeding projects. And what he manipulated was the plants that bred together. So if he had two tall plants and he bred them together, that's what he's manipulating. Or if he bred a short and a tall plant, that's what he's manipulating. Now your dependent variable is what was actually measured. Going back to that pea plant example, after he bred those plants, what kind of plants did they produce? So if you put two tall plants together, did you get another tall plant? Probably. If you put a tall and a short plant, did you either get a medium plant, which would suggest that it could be a codominant gene, or did you get either a tall plant or a short plant, which would indicate that it's a classic case of either recessive or a dominant trait that's deriving this. And that's your dependent variable. How is that response actually happening? And the big thing is you want to standardize just about everything else. Here they're calling it standardized variable. It's also just known as controls. So, going back to our pea plant example, the soil should be the same. The amount of water that they got should be the same. The amount of sunlight that they got exposed to should be the same. The time of day that the two plants were crossed should be the same. a lot of those kinds of things can be controlled for. And so that way you can't say, oh, well, the reason why this plant isn't so tall is because you didn't give it enough water. And this becomes really, really important when you're trying to do experimental design, because the last thing you want to do is have your differences that you're seeing come from something else. And again, that's your control. There's also a control group that you'll want to create. So this is an untreated group used for comparison. So in this case, that would be, we'll have to probably create a new example here. But say you have a new medication that you want to test. You'll give one group of people that new medication in either version A or version B. You'll give a second group the other version of that medication. And that third group, you're going to give them basically a sugar pill where they're not actually getting treated for something, which can bring a lot of ethical questions when it comes to human medicine and all that kind of fun stuff. So something to keep in mind. So experimental design includes these clear variables. So going back to that vaccine example, your independent variable would be the dose of the vaccine. How much vaccine are you giving? Your dependent variable would be the number of children with illnesses caused by rotavirus. Your standardized variables, so these are things you're not changing, would be the age and the health of the children in the study. And your control is the placebo lacking any active ingredient in the vaccine that you're going to give some of the kids. And again, in human situations, that can be problematic when it comes to these placebo things. So just some things to keep in mind. And I'll talk a little bit more about ethics and science a little bit next lecture, but I think it's something that's important to be broached upon in this beginning section. And again, that control is really important to make sure that, because you want to make sure that the differences that you are seeing is as a result of the medication, not just some random thing that you couldn't have possibly controlled for. Every organism is different. We all have different genetics. And so sometimes weird one-off things can totally change how things work. So it is really, really important to make sure that that control group is well accounted for. Let's get into a little bit of interpretation of data. So let's look back at this virus example here. So basically what you've got on the y-axis of your graph, you have the incidence of illnesses in case per 100 children. And then you have the dose of the vaccine. So you have a low, medium, and high dose and the placebo. And so the green represents any rotavirus illness, and the blue represents severe rotavirus illness. So here's how we're going to interpret this. If you look where those bars are, the top of the bar is telling you how many of that particular group. So say that top left for the green placebo, it has 25.86 individuals that were per 100 that had any rotavirus illness, with 14 individuals having a severe rotavirus illness. Then when you get to the low dose of the vaccine, you have roughly 2 individuals that had any rotavirus illness and 2 individuals that had severe rotavirus illness. Then by the time you get to the medium, you have 6 individuals that had any rotavirus illness with no severe rotavirus illness. And then you had almost 7 individuals per 100 children that had any rotavirus illness, with none of them having a severe rotavirus illness. So in this case, there's probably a good chance that there does seem to be an increasing effect of the vaccine when it comes to decreasing severe rotavirus illness. However, notice that that doesn't necessarily apply to the any rotavirus illness. So you may still get sick, but you're not going to be nearly as sick. So going into a little bit more specifics, in this case, the height of the bar indicates the treatment average. So if the average recovery time is in days, it's going to be somewhere between 60 and 70 for the placebo, right around 50 for the low dose, and then somewhere close to 20 for the high dose. Then these error bars that you see kind of plotted in the middle of those bar charts, that's going to indicate the variation within a treatment. Now, oftentimes you can kind of use this to really determine how different these treatments actually are. And so the bigger your error bars are, the more likely things aren't that different, especially if those error bars are overlapping with other treatments. And I could show you some real life examples at some point, but just in this case, it does look like the higher dose, which has a much smaller error bar, does seem to be much better than the typical placebo, but it may not be necessarily ruled out that it's as good as the low dose. Now, in order for us to really say some of these things, we're going to have to do some statistical analysis on the back end. So doing things like ANOVAs or chi-squared tests, which is stuff that we're not going to get into this class at all, but some things to think about. In this case, again, they did the follow-up, statistical analysis, and you see that the placebo and the low dose both are in the same group. They have that letter designation of A, whereas the high dose has the letter designation B. So in this case, it looks like the high dose actually decreased the average recovery time by quite a bit. So there's a statistically significant difference, which is good. Now kind of getting away from data analysis a bit, we're going to talk about theories. So you often hear, the phrase thrown around, evolution is a theory. That's a really poor argument because it doesn't really understand what a theory is in a scientific context. Because in a colloquial context, walking around and talking on campus, you think of theories as just, oh yeah, I had this theory that, X person liked me. That doesn't really help you very much. Whereas in science, it's something that's been, a theory is something that has been repeatedly shown to be correct over and over and over and over and over again, but isn't necessarily something that can be definitively proven. So that can be kind of complex. So, we are 99.99999% sure that evolution exists, but it's hard to say, especially given our timescales and things, that it's perfectly accurate. And, that it gets kind of complicated in that regard. There's some other theories out there. Obviously, a lot of theories are related to, and it's usually an interpretation of data. So when it comes to science, you think of scientific law, the law of gravity. A lot of these things are something where it's purely observational. And the theories are what explains what's happening in that observation. So for instance, thinking about the law of gravity, when you drop something on planet Earth, it's going to fall. But the theory is what then explains why that gravity exists in the first place. Does that, hopefully that kind of makes sense. Again, in science, a theory is an explanation for a natural phenomenon in a broader scope than a hypothesis. So remember, hypotheses are super specific. This is, in this case, much more broad and covering, say, entire, classes of organisms versus, one animal in a very specific population. Scientific theories are supported by tremendous amounts of research, and facts are repeatable in observation that everybody can agree on. So just some things to keep in mind here. I would especially focus, if you want to study well, on these bolded words, because that will probably help you study. So kind of going back to evolution for a second, when Charles Darwin, who was kind of a notorious racist, but him and Alfred Wallace kind of came up with the concept of evolution between the two of them roughly about the same time in the late 1700s. Now, when Charles Darwin saw that there were nectar tubes on specific orchids that he collected from South America, he predicted that clearly there must be a moth that had an equally long tongue, because something like this wouldn't exist if there wasn't some way to pollinate it, based off of the relationship between these moths and these orchids. So given this prediction, he then based his prediction on the fact that the theory of evolution, he would expect that there's got to be some sort of moth that's evolved to be able to utilize this resource. Otherwise, this relationship probably wouldn't exist. And roughly 30 years later, this moth was discovered, which had a super long proboscis that could allow it to get all the way down to the bottom of that orchid and draw the nectar that's in it. And, that has its advantages and disadvantages for the flower too. The longer that proboscis has to travel down that tube, the more likely that proboscis and the rest of the moth is able to pick up pollen and then move it to another flower. So some interesting things there. Scientific inquiry has its limitations. This is something that we really, really need to consider, especially with everything going on with COVID and climate change and all these other things. So some of these can include multiple interpretations. So, technically, even though we have a very strong idea that we know climate change is happening right now, there's not quite 100% consensus. There's about 97% of scientists believe that climate change is happening. Probably closer to 80-90% of those people believe it's primarily caused by man-made actions. but there's still that 3% that say, oh, this is just a natural cycle in the earth, or all those kinds of things. So there's lots of interpretations that can be made off of data based off of prior knowledge, and those aren't necessarily wrong. But those also have to be, correctly evaluated as, if there's only one paper saying that this phenomenon seems to be occurring in this weird one-off situation, and that's being cited to say that all of this stuff should be completely debunked, then that's probably something we need to consider. There can also be a misinterpretation of the observations or result. So kind of going back to our talking about controls and independent variables and dependent variables, if you set up an experiment and you didn't control for something properly, in this case, you're talking about, say, I controlled for, the amount of light my plants got, the amount of water my plants got, but you didn't necessarily control for the soil that they had. You might believe that the tall plants, when you bred two tall plants together, that created another tall plant, or a tall plant and a short plant, you bred them together and you got another tall plant. You could argue, oh, it's probably a genetic thing. However, because you gave them different soil types, it may just be that something else is going on. And that's a pretty broad example there, but there's other ways that this can happen all the time. And sometimes it comes down to just how you design your experiments. If you didn't collect enough data, so that can get kind of complicated very quickly. You also have the slow acceptance of unexpected conclusions. So weird things that happen in science aren't necessarily always the thing that everybody believes in the first time around. Feathers and dinosaurs are still kind of taking a while to become established, even though we pretty much know that there's a good chance that T-Rex was almost completely feathered. Raptors were completely feathered. However, less than 20, 30 years ago when Jurassic Park first came out, yeah, we thought they were bird-like, but it took a while for people to say, yeah, they were clearly feathered. And it took a lot of evidence to push a lot of paleontologists that direction. And finally, limited to the existing phenomenon of the natural world. You can never use science to say that God doesn't exist. Whether that be the Jewish God, the Islamic God, the Christian God, whatever you want to believe, like, you can never really use science to determine those things, because science is based off of looking at observable facts and using that to make interpretations. And that kind of conjecture is best left to philosophy and religion. So biology is constantly continuing to advance. Technology has made things very, very easy to do things nowadays. Say, for instance, some of the genetic work I do in my lab wouldn't have been possible 20 years ago, because not only have we developed new techniques that work better to get, thousands and thousands of base pairs of DNA read versus like 10. Now we can actually, use computers that are, like, say, for instance, we have a supercomputer here at UCF that helps us break down all these genetic code, and we can actually look at individual codon differences in DNA. Whereas that used to be really, really hard to pull off. And so we'd have to find shortcuts and, say, this specific gene that is hyper-conserved across everything, we know we can generate primers for it every single time. That's the only way we're going to be able to determine something's different. And that's allowed us to, have a better understanding for the actual genetic diversity when it comes to certain species, and ultimately has led to the splitting of species that we thought were the same thing. And there's a whole, that's a whole nother discussion for another time, but we'll get into that at some point as well. Another thing to keep in mind too is that pseudoscience exists. These are non-scientific claims that appear to be scientific but haven't been properly tested. You see this a lot with things like healing stones, so crystals and amber beads. Gwyneth Paltrow seems to have the market on this lately. And you really do need to be careful of these kinds of things. Essential oils. I'm actually very, very annoyed with the whole thing of essential oils. Until you can show me a scientific study that has been peer-reviewed that shows that essential oils actually do something other than just help with general relaxation, yeah, they're not curing cancer, guys. Sorry. But that's something to keep in mind as well, that does exist out there. If y'all are big podcast fans, there's a podcast called Skeptoid that does a really good breakdown of how to spot pseudoscience. That's really useful if you've ever had to watch the Discovery Channel or the History Channel and watch some of their weird cryptid shows where they talk about Bigfoot and Loch Ness Monster, or the weird stuff that shows up sometimes around Shark Week, where megalodon still exists out there, and it's all crap. But I mean, it's entertaining for some, but it's all crap. So it is really important to make sure that we're not letting pseudoscience kind of creep in there. So claim is pseudoscience if it seems scientific but is not based on a testable hypothesis. So say, for instance, just because we haven't found megalodon doesn't mean it's not out there. That's a really bad hypothesis. You can't use that. It's never been tested experimentally. Again, going back to the healing stones or water has memory or any of the weird theories that have popped up over, say, the last 40 years. As well as it has been tested experimentally, but the data does not support the conclusions. This can happen often with animal behavior. Unfortunately, there's a lot of conjecture about how intelligent an organism really is and whether or not it should be held in captivity, as far as zoos and aquariums and all that kind of fun stuff goes. And there's quite a bit of that realm of unfortunately a lot of individuals coming more from the psychology aspect of it that think about humans and anthropomorphizing animals, and it doesn't quite work. And so it can be problematic, to say the least. So the big thing is you really want to make sure you're evaluating sources. So probably the first one you think of that'll probably be pretty strong is textbooks. That's not 100% true. Obviously, with this coming from the McGraw-Hill Education Group, they're most inclined to say textbooks first, but you know how that goes. But in theory, most textbooks should be pretty solid. Websites. Anything that tries to sell you something is probably going to be really on the lower tier. So, some of these websites that claim the healing powers of healing stones are probably the same ones that are usually trying to sell them to you. Then you get to personal and social media websites, so blogs, vlogs, all that kind of fun stuff. Institutional sites without references. So these are often like, for instance, there's a guy named Raymond Hoser in herpetology that has gone out and we call it taxonomic plagiarism. It basically means that he went out and assigned names to things that shouldn't have gotten names. And so he has his own page. He makes it look all fancy, but none of it's peer-reviewed. Nothing is good. Then you have sites with references. So I'm actually not the biggest proponent or biggest anti-Wikipedia person, because honestly, more often than not, it's more accurate than some of these other places you're going to see on the web. So it could be worse. And then you're going to finally get to my personal favorite when it comes to evaluating whether something is factual or not, an actual scientific journal where the research has been properly peer-reviewed, which even then isn't always the case. There are examples of academic journals slipping up and letting something through, and it can damage entire fields for 30 or 40 years. The vaccine autism case is a very particular one where a study that was not properly peer-reviewed got through, published in a major journal, and was retracted within a month. But now we still have a lot of people out there that believe that vaccines cause autism because this particular scientist basically misled the world. So it has made quite a lot of money off of it too. So again, coming back to, if it's trying to sell you something, that's probably a bad sign. So here's 5 questions that I want you all to ask whenever you're evaluating a scientific claim. And again, this is really, really useful when you're thinking about COVID-19 stuff right now. There's a lot of misinformation kind of flying around. There's a lot of stuff that we've learned since May that doesn't line up with what we thought in March, all that kind of fun stuff. So the first thing is, does it seem logical? Now, if it seems to check all of your boxes 100%, you should probably double check that too. If it seems too logical, there's a chance that maybe it's answering one of your own biases. Is the source credible? Is it from somebody that spent 30 years getting a PhD and then starting their own lab? Or is it somebody that's just posting on Google? Mom blogs in particular are kind of detrimental in this regard. Are the scientific studies cited? In other words, if it's referring to studies, are they the correct studies to be referring to? Do those studies actually say what this new bit of information is saying that it says, all that kind of fun stuff? And then you want to look at, are their experiments well designed? And this is stuff that as grad students we go through, and whenever we read a new paper, we're looking at the experimental design, not only to see how we can do very similar things, but to kind of get a better idea on those sorts of stuff. And then finally, does the data match the conclusion? So of their data, not only did the previous stuff that they pulled in from other stuff seem correct, but does their data and the conclusions that they're drawing seem correct? And again, that's stuff that, even peer-reviewed science doesn't always catch. So here's another great video that talks about scientific method and interpreting graphs. I'll leave that link in the PowerPoint so y'all have access to it, because at this point we're kind of probably running a little bit long here. So with that being said, let me go ahead and stop screen sharing here. I hope y'all enjoyed your first lecture. Again, this is an asynchronous class, which means that, y'all are more than welcome to keep watching the next lecture right after this, or, take a day or two break, process this information, and start the next lecture. So however y'all feel that it makes the most sense to do these things is totally up to you, and I hope that you're able to find success in this course, because it's going to be a little bit stressful this semester than it normally is. So with that being said, I hope y'all have a great afternoon and have a great rest of the week.